Yeah, it's interesting too, because like, you know, in Washington, for the preservation of a lot of those populations, to have any type of fishery, I think commercial, recreational, subsistence, cultural fisheries, so depend on these hatchery systems. And it's just really complex because we want to like revalue these natural wild populations so that maybe we can realize, okay, this past
I sure you someone in one man, just
maybe we can focus on that bonus tradition.
Okay. Okay. I'll do that.
But then you still get to that point. Yeah. Like up here, you still have like the opportunity to do it. But it's like it's almost like, well, it's sort of gone past.
Hey, Julie.
Yeah.
Can you bring it back and then, yeah.
Yeah. I feel like it's like really basic.
Very basic.
And yeah, that's not really questions.
I honestly want to see what I got over the last one before I like
Want to make it harvest it, make it small, like, spaces, energy, and that build the freezer. So we can take the bench as well, success, like, we want to have this system, access to as much as possible. Like, 40 was fast, you're not going over the bench. Versus, the opposite perspective of that, perspective.
Okay.
I don't know how to run Windows 10, so I can't find my jump drive.
Oh my god, dude.
Yeah.
Well, normally on mine it shows up over here.
On a computer section? Press this thing.
Yeah, tell
That
me.
means I'm not gonna be able to find mine either. This is bad.
We'll find it.
We'll find it.
Isn't this?
Ooh, press the
there,
thing, press the thing.
That took like 5 minutes. There, okay, there's, oh, they both showed up.
Okay, mine's full drive.
Good. You want to open yours?
Yeah.
This one? 1259, not 1257.
Yeah, because like
Yes.
me.
That one's a PDF in case the PowerPoint fails.
I'm thinking.
I was sitting in my office trying to review all of Bayesian statistics and Markov chain Monte Carlo.
No way.
I was like, okay, well, this is as good as this is going to get.
Yeah, I was just gonna say.
Yeah. We're gonna go out into another class today.
Evan's back too.
Okay.
Hello.
Hey, Brooke.
How's it going?
Good, how are you?
Oh, pretty good. I'm taking one history class this semester.
Oh, okay.
Minority.
How's that going?
This one? Or...
Yeah, well, the fisheries class, good.
Oh, it's good. Yeah,
Good.
it's important to see the human and cultural dimension.
Yeah. Oh, were you taking Courtney's class?
Yeah.
Oh, nice.
Yeah.
Good for you. Good deal. How was your summer?
That's awesome. Busy. We had the couple at home, so I had a lot more responsibility.
Oh, okay. Oh,
It was good though. I learned how to drive the
oh that's good. Yeah.
All good stuff.
Good.
Long hours and then responsibilities as like parents, cooking, and they want to go swimming, but they need adult supervision, you know, stuff like that.
Yeah.
It's good though, it's rewarding.
Yeah. Good.
Yeah. How about you?
Well, you know, summer always goes by too fast, and then it ends, and you're like, where the heck did summer go?
Yeah.
So that's the way I felt, but it was good. I went back and I did 2 weeks of field work, and then was here for 3 weeks, and then went back and visited family in Maine.
Oh, nice.
Yeah, and then was back here.
Sorry, it's October.
I know.
Sad.
Yeah, it is. So.
Okay, good to see you.
Good chatting with you.
Andy, can you hear me?
Andy? I guess not. That is 201.
Nice to see you here. How's it going?
Remember the Esther table?
Did you not get a permit?
Where'd you park?
I think if you have a department vehicle,
Just never know.
I feel pretty low, bro.
Marguerite, can you hear them?
Hello, this is Scott with Video Conferencing Services. For the people on PEXIP, I can hear you just fine. Juno, I can also hear you just fine. For the Fairbanks room, are you able to hear all of your distance participants? You are currently muted. Okay, I'm going to be giving a call to the text there.
Hey Julie, what's up?
Should be like oil in it.
You know that we couldn't hear you, but
I can't hear
now,
Julie, for some reason. I can see you talking.
well, I
Still
think
can't
you're.
hear you.
There's
I'm
a
gonna get a hold
problem.
of the IT people in
Nope,
CFOS
can't hear
and
you.
get that resolved.
I have to reboot it.
Christina says you might have to reboot PEXIT.
I can hear Julie in Anchorage.
Oh, there she goes. Hi Margot. Can you talk, Margot? Can you unmute yourself to see if it works?
Yeah.
Her volume
Yeah.
down? No.
Nope.
Can you hear
Okay, say
me?
something.
I am saying something.
You are saying something? No, I can't hear you.
You can't hear me?
I think they have to reboot.
Okay, Christina's gonna fix things because she's amazing.
Okay, and so this is Juno.
Jump out of the thing.
Andy, are you guys in presentation mode? We can't see a presentation.
It's all in these statistics.
This is the word deck area, like undercover or something.
It's very funny in back there.
Yeah.
Gives an itch now.
Then it's an infinite.
Hello, this is Scott with Video Conference Services again. O'Neill, are you able to hear me?
On.
You should be unmuted.
Can we sound check with the remote site, please?
I can hear you guys.
Oh, we got you, Margot. Cool.
Awesome. I disconnected and reconnected. I think that worked.
So in Juno, we can hear you, but we can't see a presentation.
Okay, we haven't gone into presentation mode yet, so that's okay. Thanks, Juno.
Okay.
We don't have a presentation up, Andy.
Well, they can still see.
They can still
we
see
should
the
be able to
screen.
see the screen.
Okay.
And then we can.
Oh, beautiful.
Can you see the presentation now, Milo? Hey, team. Demonstration.
Margot says
Magnifying glass.
No, we can't.
Really?
There's nothing coming through the projector. It's all on frames on the TV.
You get presentation?
Well, we did.
Hi Juno, this is Scott with Video Conferencing. Could you check to make sure that that projector's on? We're able to see the content at all the other sites.
Yeah, the projector is on. It's not projecting anything.
Okay, I'm going to disconnect you real quick and bring you right back into the conference. Hopefully that will resolve it.
All right, thanks, Scott.
So I walked up here and realized I had a pen light and not a pen.
We didn't quite do it.
Okay, we're going to get a hold of Chris Brooks and see if he can help you guys real quick. You should be seeing it.
Sorry, Scott, I missed that.
We're going to give Chris Brooks a quick call. You should be seeing the presentation. Hopefully he can
Okay,
get down
thanks.
there quick and help you guys get that working.
Limelight. We'll wait until Juno gets there. presentation up.
See them all go into the guy that's like there's not range this big before they It was established.
Oh, really?
Hey Fairbanks, this is Chris Brooks. Can you guys stop your presentation real quick?
Stop it?
Yeah, stop it.
Okay. Okay, I stopped it.
Alright. Okay, just give us a second here. Okay guys, you can start it again.
Okay, I started it. That was exactly like
I have like You have to sign up for the one. Yeah, that's basically what I got. I signed up for the CFOS. They're all like,
This is where
can they see the pictures? Do like all the
So,
what happened to power cycle and stuff? I mean, practice, so practice.
Point the camera at the screen and go. No, serious.
We told them to go ahead.
Fairbanks, we're going to go off for a second, and we're going to have to reboot real quick. Yeah, we'll be back with you.
okay.
I only have one fish joke, Yankees.
What?
I'll share it. Do you want to, does everyone want to share? No, do you want to hear my fish joke? Let me hear you. What did the fish say when it swam into the wall?
Sure, my goes in. Damn, that one again?
I told you I only one fish joke.
I haven't heard of it.
So don't try to think Spanish doesn't work. Oh, actually. And then you have to sit there and try to explain that in English.
The Margot clearly is not
Well, right, what happens? It works with me.
I don't have
Gather around.
link it on the
Just link it right on the computer.
Yes, in Juno. Is Juno?
No, Juno is the one place where we're having it, so every single day.
What time?
We just use somebody's classroom.
What time are you fish tank? So somebody unplugged the cable.
Yeah, but they're also here.
You ready to go, Juno?
We're just getting into the conversation.
Andy, we had to switch rooms, but we're ready.
Okay. All right. I'll start. So, and I'll keep my introduction fairly brief. It was about 20 minutes long. So it is my pleasure to introduce Kaitlyn Manishin. She's defending her master's thesis today. Kaitlyn grew up in Half Moon Bay, California. And I put together a little slideshow for us, just so we can picture things. And this is the idyllic version of Half Moon Bay, but that's what the Tourism Bureau tells you it looks like, but this is what it really looks like. And notice the fog in there. And Kaitlyn, Half Moon Bay is most well renowned for being the home of Mavericks, which is one of the biggest surf breaks in the world. And that's actually a picture of Kaitlyn that's floating around the internet surfing Mavericks during a big wave competition there. So Kaitlyn decided to go to college in the one place in the world that's even foggier than Half Moon Bay, and that is Humboldt State, where she majored in fisheries biology. Is that correct? Yes. Yeah, with the marine option. And this is a double Kaitlyn, because there's a Kaitlyn and there's a Kaitlyn. So she was apparently the poster child of the program, too. And she also minored in applied mathematics and applied statistics. She took a couple years off, 2 or 3 years up, 2. 1.5. After her undergrad on an ambulance crew as an EMT. And she's also worked as a wilderness leader at kids camps, and while she was at Humboldt State, she also worked as a statistical consultant type person for the Henry Spork Foundation between undergrad and grad school. And then there's a bunch of other stuff that she did too, but I won't bore you with those details. She moved up to UAF, and apparently she really liked operating power tools. So she also operated a chainsaw. And she liked Moscow mules, apparently, made some good friends along the way, and liked hoodoo beer. And I've reused this picture like 5 times now, but pictures of people wearing goofy holiday costumes are pretty fun. And then notice this is called a Kaitlyn sandwich. So there's a Kaitlyn, a Marguerite, and then a Kaitlyn. And this is further exemplified in this. There's another Kaitlyn sandwich. And while she was operating power tools and sandwiching Marguerite, she actually managed to get this great thesis done, which she's about to tell you about today. This is Kaitlyn presenting her work at the Alaska Marine Science Symposium. And then I'd like to note another couple things. She just recently passed her EMT2 certification, so she is a great person to have around in case you stub your toe or get a hangnail. And she is also graduating with a graduate certificate in statistics, which I'll put a plug in for all you fisheries master's students or PhD students. It is a great thing to do because you end up taking a lot of statistics courses, and you get this nice little footnote on your diploma, and it looks good to potential employers. And Kaitlyn cruised through that program and did really well. And with that, I'll turn it over to Kaitlyn. Maybe. Oh. There you go.
Hi guys, so that was significantly less embarrassing than I thought it was going to be. So that's good, and that's statistically significant. So today I'll be presenting what I've been doing for the last 2 years, which is exploring the potential role of late-stage predation on snow salmon demographics. And I would like to acknowledge my committee, Craig Cunningham, who's joining us in Juneau, Peter Wesley, Ken Goldman, who couldn't be with us today, and my advisor, Andy Seitz. So my thesis is structured into 2 chapters. So today I'm going to give a general introduction before diving into chapter 1, which looks at per capita prey consumption by salmon sharks. We'll go over methods, results, and discussion for that chapter before we move into chapter 2, which looks at that late-stage mortality and its impact on Chinook salmon age structure. So we'll go over methods, results, and discussion for that chapter before going to some general conclusions. So to dive right in and introduce my focal species, for those of you who don't think about salmon all the time, I'm going to be studying, or I studied, Chinook salmon, Oncorhynchus tshawashaw, which is the largest and least abundant of the Pacific salmon, and it's a very important cultural and ecological resource throughout its range. It has a long history of exploitation, but in recent years it has expressed depressed abundance and productivity. So in Alaska, Chinook are generally what's called stream type, where they spend 1 year in fresh water before heading out to sea, where they can mature and grow, generally for 2 to 4 years, but sometimes as few as 1, as many as 5, before returning to their natal stream to spawn. So it's been observed throughout the range of salmon that they've been getting smaller when they return to the river. So this was first noticed in 1981, was reported again in 1996, and zooming back in specifically to Chinook salmon, Bert Lewis and co-authors reported in 2015 that Chinook are getting smaller and younger at return. That was in Alaska, and this was expanded throughout the range of Chinook salmon, which is from Central California up to Alaska, by Jan Olberger and co-authors in 2018. So you have to remember that when salmon return to rivers, they return at a variety of ages. So when they're coming back smaller, this could be due to 2 reasons. It could be due to changes in age structure, like getting younger, or changes in size and age. So some work out of the state of Alaska Salmon and People Working Group looked at the proportion of total change in length due to these 2 factors. So here we've got change in length on the y-axis and populations of Chinook salmon on the x-axis. The black represents the proportion of change due to age structure, where the yellow represents the proportion of change due to size and age. And you can see that there's a lot more black than there is yellow. So a lot more of this change in length is due to Chinook salmon getting younger than it is them getting smaller at a given age. So these type of demographic changes have been reported as a result of top-down pressure, for example, commercial fishing. And an example from the east coast of the US and Canada on Atlantic cod. This is a very simplified picture, but on the left panel we have before fishing. You've got your oldest and largest individuals present. Fishing tends to be highly selective, so you remove those oldest and largest individuals, and then the ones that are smaller or younger are left to pass on their genes, and after a round of reproduction, the population is generally smaller and might mature earlier. So an alternate source of top-down pressure is from predation, and this has also been shown to have similar effects on demographics. Here we see an example from Trinidadian guppies, where 2 populations of guppies, one that experience high predation, one that experience low predation. So the populations in high predation environments mature earlier than those in low predation environments. So, given the demographic changes in Chinook salmon and these reported causes of change in other populations and the geographic extent of the changes in Chinook salmon, our lab wanted to examine Chinook during their ocean phase. So we put pop-up satellite tags on large and mature Chinook salmon out of Dutch Harbor, Alaska, and got some pretty unexpected results. So this is a time series from one of those tags, and I'm going to walk you through it. On the left y-axis is depth, represented by the black line. On the right y-axis is temperature, represented by the blue line, and the x-axis is date. So if we follow the fish starting on December 18th, the fish is swimming up and down in the water column, experiencing an ambient temperature in the Bering Sea of about 6 degrees Celsius. On December 27th, all of a sudden the temperature jumps up to over 25 degrees Celsius, and the fish continues moving up and down in the water column. So the other thing this tide recorded was light, even though it's not on this plot. And during that temperature spike, the tide recorded complete darkness for 3 days, even though it was showing day and night alterations before that. So what in the Bering Sea in December is 26 degrees Celsius, completely dark, and moving up and down? We decided that the inside of a salmon shark was that place. So salmon sharks are an endothermic shark species, so that means they retain some of their metabolic heat and maintain a temperature differential with their ambient environment. So these fish are a bit warm-blooded, around 25, 26 degrees Celsius, which is cooler than a marine mammal and warmer than a cold-blooded fish. So they're just about the only thing in the Bering Sea that's 26 degrees Celsius, especially in December. So the other surprising thing about this study was that this pattern, this temperature spike indicating salmon shark predation, was observed in 14 of 35 tags that have reported their data, and temperature and depth patterns indicating other types of predators were seen in an additional 3 tags. So that's pretty high incidence of mortality. So due to the changes in the age structure, observed changes in the ages of Chinook salmon, they're getting younger, and this tagging, we adapted this hypothesis that I'm going to call the dangerous ocean hypothesis, which is could increased selective mortality after the first ocean winter have resulted in these changes in age structure in Chinook salmon. So basically, if there's more mortality risk in the ocean, that risk could be a selective pressure on the salmon, say, causing them to mature earlier than before. Additionally, Chinook salmon would be the most susceptible to this pressure of any of the other salmon species, since they spend more time in the ocean maturing and would be subject to that pressure for longer. So in order to tie together our salmon sharks and our salmon, a logical way to do this is to look at salmon shark per capita prey consumption and scale that up to the population level using the proportion of Chinook salmon in the diet and total abundance of salmon sharks, and plug that into a population dynamics model to see how it affected salmon populations. The problem with that is when you start looking at the data, those diet proportions for salmon sharks and their total population abundance don't really exist. We don't have that data right now. So instead of addressing this problem in a one-way linear fashion, I look at it as I'm addressing it from both the predator end and the prey end. So my first chapter is from the predator end, and I'm just going to look at the per capita total prey consumption by salmon sharks. So how much prey does one individual salmon shark consume? In the second chapter, I look at how much theoretical additional mortality would be required to see the changes that we've observed in Chinook salmon populations. So with that, I'm going to take a sip of water, and then we're going to move into chapter 1. So my first chapter is titled Prey Consumption Estimates for Salmon Sharks. I should note that Margaret Short from the Statistics Department is also a co-author on this chapter, and it's in review with marine and freshwater research. So to introduce the focal species of chapter 1 is the salmon shark, Lamnoditropus, a member of the lamnid family. Other members are mako sharks and the well-known white sharks. So all of the sharks in this family are endothermic, but salmon sharks are the most endothermic. They've been observed to maintain a temperature differential of as much as 24 degrees above ambient, while short-fin makos maintain a maximum of 8 degrees above ambient, and white sharks maintain a maximum of 14 degrees above ambient. Salmon sharks can grow to be fairly large, reaching over 200 centimeters pre-caudal length as adults, and can live to be over 30 years. Though their diet is not well known, they're thought to be generalist, opportunistic predators, eating a wide variety of prey. So the salmon shark population exists on several gradients, with males dominating the Western Pacific and females dominating the Eastern Pacific. Additionally, the females make a patrician migration down to the southern end of their range, so they pup down at the southern end of their range, and then the juveniles slowly migrate north as they grow. That makes most of the salmon sharks in Alaskan waters sexually mature adults. So salmon sharks are actually a fairly well-studied species for a shark, but being a non-commercial species, there's still those large data gaps that we mentioned. So to address this problem, I'm using 3 separate estimates to calculate salmon shark consumption, using the agreement or disagreement between these estimates to make for confidence or lack of confidence in my results. The first is the daily ration requirement, which is a % body mass per day. I use this as a data light method to get a ballpark idea of where consumption might be. The second method is a bioenergetic mass balance, which sums up all of an animal's energy expenditures to calculate how much energy that animal needs to sustain itself. So this is good for animals where directly measuring things like food consumed versus waste excreted is impractical, or for aquatic organisms where measuring things like total heat produced is not feasible. However, this method requires a large suite of biological and life history parameters, so it's pretty data intensive. My final method uses the fitted parameters of a growth model to calculate consumption. So this attempts to eliminate those data requirements of the mass balance by only using age and length data. However, it has been tested in endothermic tunas, so a somewhat physiologically similar species, but has not been tested in lamnid sharks. So to dive into the methods, the daily ration requirement, percent body mass per day, has not been calculated for salmon sharks, so I used ration requirements from closely related species. Specifically, I borrowed from shortfin mako sharks, who range from 3% body mass per day to 4.5%, and white sharks, which range from 1.2 to 1.5% per day. So I resampled those percentages from related species to get a new mean, and applied that to the average weight of an adult salmon shark. In the bioenergetic mass balance, I summed energy towards active metabolism, growth, reproduction, digestion, and excretion. So salmon shark specific data went into active metabolism in the form of swimming speeds, growth from a growth model, reproduction in the form of pup length, average number of pups, and length of reproductive cycle. And then digestion and excretion were simply proportions of the total, and those were borrowed from other species. So unlike the other two methods, the mass balance outputs in terms of energy, not in terms of biomass. So in order to compare with the other two methods, we have to convert energy to biomass. To do that, you need to multiply it by the energy density of the prey. So this presents a bit of a problem. As we've already discussed, the salmon shark diet is not well known. So what I had to do was take few observations of few individuals and take those stomach samples and extrapolate some diet scenarios. And so I created these scenarios and I had them vary by the amount of salmon in the diet based on some seasonal observations of salmon sharks. So I ended up with a high salmon diet, which is meant to mimic the summer diet and results in 3% of Chinook salmon in their diet. Then a high medium salmon diet, which is 1.5%, low medium salmon diet, which mimics their fall diet for 0.5%, and a low salmon diet based off the spring and winter observations with 0.25% of Chinook in the diet. So that brings us to the final method, which I used a generalized von Bertalanffy growth curve fit using Bayesian, hierarchical Bayesian methods to calculate consumption. So before everyone has a conniption and just falls over, the Bayesian part means that I use prior knowledge when informing the fit of this model. So in this case, I used parameters from a non-generalized form of the von Bertalanffy growth curve fit by Ken Goldman and John Musick in 2006 as a starting point for fitting the model. The hierarchical part means that I treated individual as a random effect and fit a growth curve for each individual and used the mean of those growth curves as an estimate of the population level growth function. So once we'd gotten through the growth model fitting, I then calculated consumptions from some of the fitted parameters. So consumption in kilograms per year was equal to H over A, which is the assimilation constant, or just the constant that scales weight into consumption, over the assimilation efficiency, which is the percent of available energy absorbed from a prey item, multiplied by the weight raised to 2, plus the allometric slope of consumption, which is how consumption scales with body size. So H and D, allometric assimilation constant and the allometric slope of consumption, were both calculated from fitted model parameters, but A was borrowed from some other species. So now getting into the results. On the y-axis is consumption in kilograms per year, and on the x-axis we've got the model method. So these are box plots with the center line being the median, and the outside lines, the hinges, being the 25 and 75% percentiles. Yep. And the whiskers go to 1.5 times the inner quartile range. So you can see pretty clearly that we've got two methods that are fairly close to each other and one that's way different. So from the daily ration requirement, we got an annual requirement of 1,500 kilograms of prey per year. The bioenergetic mass balance output 8,580 megajoules, so that's in terms of energy, and then depending on the diet scenario we use, that gave between 1,800 and 2,100 kilograms of prey per year. Our final method, growth model, gave 17,000 kilograms of prey. So now I'm going to dive into some specifics about each of these methods before interpreting the results in a bit of a more ecological context. But before we do that, I would also like to note that, so here we've got consumption again on the y-axis, and this is diet scenario just from the mass balance on the x-axis, and all the diet scenarios were fairly close to each other. So at least for this exercise, that diet scenario assumption didn't make too much of a difference, so it's fairly comparable. So now moving on and looking at each method. So if you remember, the daily ration requirement only used information from closely related species. So this method should really be used as a ballpark estimate for a salmon shark-sized lamnate shark. So this is like a general starting point. And then diving into the mass balance method, here we do start to incorporate some really salmon shark specific data in swimming speeds, growth, and reproductive parameters. However, some of the relationships and constants that we use to transform those values into energy were borrowed from other species and sometimes from juveniles of those species, and some of those relationships might not hold for salmon sharks or might not scale linearly with size. So in conclusion, we use a lot of good salmon shark information here, but there are some places in the calculations that could bias our results. And finally, the growth model, obviously very different from the other methods, used a lot of salmon shark specific data. We used salmon shark age and growth data, except we still had to borrow those assimilation efficiencies. And it was expected, based on previous work, to be unbiased for fish with decelerating growth rates, which salmon sharks have from that model plot that I showed you before. However, this method has not been validated in lamnid sharks, so it's not been fit to a shark that has a reliable consumption method and then compared to a method that has already been established. So now moving on to some more ecological context. If you scale closely related species down to the size of a salmon shark, a white shark would consume 800 kilograms of prey per year, while a mako shark would consume 2,200. And our minimum, most conservative salmon shark estimate is 1,500 kilograms of prey per year, which falls right in the middle of those two related species. So something to note about that is that both makos and white sharks are bigger than salmon sharks, so that results in them having a lower mass-specific metabolism. Additionally, salmon sharks maintain that much higher thermal gradient with their environment, so those two facts indicate that salmon sharks may have higher energetic requirements than those other species. So what if we compare salmon sharks to something like a killer whale? So if you scale a killer whale down to the size of a salmon shark, you end up with it requiring 11,000 megajoules of energy per year. And from the bioenergetic mass balance, we got 8,580 megajoules, which actually aren't too far away from each other. So this corroborates the findings of some previous authors that the energetic requirements of endothermic fish could be somewhat similar to those of birds and marine mammals. So obviously this study has a lot of room for future work, specifically in salmon shark specific biological parameters, such as metabolism, which is challenging to study in larger animals, but in the picture on the right, you can see a juvenile short-fin mako shark inside a respirometer, which is used to measure its gas exchange. Then salmon shark specific energy density from bomb calorimetry. You could get an assimilation efficiency from captive feeding studies, and finally, a model fitting exercise with that phased growth model method to see if it's a valid method to use for salmon sharks would also be helpful. So some general conclusions from chapter 1 is that this suggests that energetic requirements of salmon sharks could be similar to those of marine mammals, and that means they could have a potentially large impact as an apex fish predator. However, these conclusions are highly uncertain and much more data is needed. So if I lured you guys with the how many salmon do salmon sharks eat on the poster, then I totally duped you because I'm not going to tell you. Okay, and that brings us to chapter 2, which is late-stage marine mortality of Chinook salmon may contribute to changes in age structure. So let's bring it all back to where we started with these tagged Chinook salmon. So these fish were tagged in Dutch Harbor in the Bering Sea, which made us want to look at Western Alaska populations. So we chose the indicator population of the Salcher River, which is a tributary to the Yukon, a major river in the region. Additionally, the Salcher produces 9%, almost 9% of the adults returning to the entire Yukon drainage, and it has a breadth of data available and has been subject to some past studies, which I capitalize on. So I use a model established by Cunningham et al. In 2018 and run simulations with that model to compare age structure to that collected and reported by the Yukon Joint Technical Committee. So to introduce this model a little more, it's a really exciting tool because it incorporates both top-up and bottom-down drivers of Chinook productivity. So it incorporates the top-down drivers of bycatch and harvest and some bottom-up environmental covariates. However, it does not explicitly incorporate any predation, so that's where my study comes in. I'm going to put in different scenarios with different levels of predation. Additionally, I should note that in the model, the probability of maturation is held constant in each year. So to walk you through what this model actually does, fish start in this upstream environment, and they then transition to their downstream and nearshore environment before moving into the ocean. Once in the ocean, fish have a bunch of options. They can either continue on in the ocean, end up as bycatch in another commercial fishery, or mature and return to the river. Once in the river, they're subject to terminal fisheries or terminal harvest, or they can continue on fully and complete their life cycle. So all I'm doing is adding another option to that marine phase. Throughout running simulations with this model framework, the number of life stages such as eggs, upstream juveniles, downstream nearshore juveniles, ocean ages at each stage, my predation removals that I've added, and total adult returns are recorded in each year. So we keep track of everything that's going on. So what I want you to take away from that is in order to apply additional predation in the ocean, I have to do it at 5 different stages. I have to do it for each ocean age. So that brings us to simulation scenarios. So this is where we get a little complicated, but it's okay. So I wanted to look at both the selectivity and the total pressure of predation. So I have 14 different selectivity scenarios, which are on the y-axis here. They're divided out by ocean ages 1 through 5. So when you see bigger, darker dots, that means more additional predation is being applied to that age class. Smaller, lighter dots mean less. I did this in 3 phases, which each phase of scenario proposal iteratively informing the next phase. So that's a bit of trial and error. In the first phase, I was putting extra mortality into the model really because I wanted to see how it behaved, so this was pretty experimental. In the second phase, scenarios 5 through 10, I was trying to influence that age structure to see if I could get some of the observed shifts. Towards the end of that phase, I noticed that predation on certain age classes was helping me reach that age structure, and so I focused on those age classes in phase 3. So you guys could be looking at that rate and wondering what the heck do those numbers mean. So those are in log space and transformed back to a total annual predation rate. The lowest I applied was 0.5 and up to 63% additional predation. So that's the selectivity part. So what about that total intensity of predation? So I scaled each of these scenarios by a scalar from 0 to 2 by increments of 0.2. So that means I just moved them up and down. So I went all the way from 0, so that cancels out additional predation, so basically a null model, to 2 times what you have here. And when you translate that to percentages again, that means my minimum percentage applied was 0 and my max was 83% additional predation. So I had some pretty good coverage over additional predation. So then we get to the problem of trying to pick which scenario was best. And so I told you before, we're comparing the simulated model output with this observed age structure. So on the left, we've got the baseline model output with proportion escapement on the y-axis and year on the x-axis from the model with no additional predation. And we've got the observed age structure on the right. So each chunk of color represents one ocean age class. That blue bar towards the top is ocean age 2. The tealy color in the middle is ocean age 3. And the green towards the bottom is ocean age 4. So you can see this trend is a little noisy. So in order to pull out the trend, we did some multinomial modeling, which gives us the probability of a fish returning at a given age in a given year. So that's what we did to pull those trends out, smooth things so that we could see better what's going on. And then we have the second challenge of comparing these two, which is that they cover different years and they have different numbers of years. So in order to overcome that challenge, we decided that we are going to compare the most recent common year between these two time series. So to do this, for each scenario, I calculated the likelihood of the age structure from the scenario given the age structure in 2010 from the observations, and I ranked those log likelihoods, which resulted in this plot. So these are all the log likelihoods divided by phase, and this top 30, this is the top 35, are all from phase 3, which, if you remember, were very selective against ocean 3 fish, and the last 2 scenarios in phase 2, which were also very selective against ocean 3 fish. Then these very top 9 scenarios, those are all from phase 3, so all selective against ocean 3 fish. And our top scenario is 11-08, which means selectivity scenario 11 scaled by 08, and I'm going to explain to you what that means. So if we go back to selectivity scenario 11, it only applied additional predation to ocean 3. So only ocean 3, no other age classes. And from tracking through the model, we found out that the average additional mortality ended up being 47%. So we applied each year 47% additional mortality at only ocean age 3. And that allowed us to more closely match the proportion escapement from our model with the observed proportion. And so here on the left is the observed, on the right is the model, and they look pretty similar, which is what you hope, because this is what our method selected as the best. So we match the age structure, but what happens to the overall returns after we've put in this extra predation? Is this realistic? So as I mentioned before, we also tracked overall returns. And here we've got adult returns on the y-axis and year on the x-axis. That purple line is the base model fit to the data with no additional predation, and that red ribbon is the 95% Bayesian posterior, Bayesian credible interval, sorry. So when we put in our leading scenario, it falls outside that 95% credible interval. So that means it's really not likely that fish were returning in that low of abundances. So what this means is the simulations recreated the age structure, but we weren't able to recreate the age structure while maintaining a realistic level of abundance. And something to remember here is that this model, this tool that we use to run the simulations, uses a fixed maturation schedule. So even though we're putting in more mortality late in the ocean phase, it doesn't make the fish likelier to mature before that mortality would happen. So this indicates that a maturation schedule shift could have already occurred, Because if that had already occurred and sufficient maturing earlier, then you would need to remove fewer individuals to see the same magnitude in age structure change. So you would get the same age structure shift with less of a decline in overall returns. So some other discussion points from this chapter are that we predicted intense selective predation on ocean 3 fish, and there also have been some observations of this occurring in nature. So killer whales tend to consume ocean 3 Chinook more than other age classes, and it just so happens that all the fish that were tagged were at the end of their ocean 2 year and beginning of their ocean 3 year. So additionally, our top scenario resulted in 47% additional mortality, and if we look at the numbers from our tagging study, 40% of those were eaten by salmon sharks. And if we add in other predators, 48% succumbed to predation, considering all those events. So though this tagging study wasn't designed to measure mortality rates, it's a really interesting alignment of this opportunistic observation with a totally independent model simulation. So in conclusion of everything, salmon sharks might eat a lot of prey. That's highly uncertain at this time. Top-down pressure on ocean 3 Chinook could drive age structure, but we can't reconcile these two pieces of the puzzle, these two ends of the equation, with the data gaps that currently exist. So future work from this study would obviously be salmon shark diet and population abundance estimates, and then getting marine mortality estimates of Chinook salmon for all age classes. I'm sure a lot of modelers would really like that. And then running a population dynamics model, which includes the evolutionary effects on maturation. So finally, I have some acknowledgements. I would like to re-acknowledge my committee, this time in order of pictures with dead things. Because that's what you see when you hang out with these guys. And then also the Sites Lab, Marguerite Tibbles, Michael Courtney, Julie Nielsen, Matt Smukel, Thomas Ferrugia, and Steph Meggers. Those guys have heard how much salmon sharks eat so many times, it's not even funny. And I would also really like to acknowledge my Chapter 1 co-author, Margaret Short, who really went above and beyond as a professor and helping me out with the Bayesian statistics component. On a personal level, I would really like to thank my family. The last couple years have been quite a challenge, and the great grad student community here at UAF, especially the ladies of the outhouse, along with previous mentors who I often called on, the Esther Volunteer Fire Department, because I couldn't have got through without some other outlet from you guys. Danielle Garrick, my longtime roommate who saw me through the worst of it, and then finally everybody else's pets. With that, I thank you all for sitting here and listening, and I'll answer any questions.
So ask the distance sites first.
Right, okay. Distant sites, do you have questions?
I have a question in Juneau. I'm just curious how you got to the idea that the salmon shark's metabolic demand could be approaching that of a marine mammal, just because the sharks aren't whole body endotherms, right? They're mostly heating their gut, and it seems like living this far north, yes, they have to heat their gut a lot more than the ambient temperature, but then the rest of their body that's not heated is saving quite a bit by living in such cold temperatures. And then, I don't know a ton about this, but I would expect that marine mammals have a much higher metabolic their brain, because they have so much more complicated social structure and so on.
So that conclusion came out of comparing the annual energetic requirements between the killer whale and a salmon shark. When you scale the killer whale requirements down to the size of a salmon shark, since they're a much bigger animal. So even though salmon sharks keep mostly their viscera and their, I think, brain and optic nerves pretty warm, even though they only keep those parts warm, they still have that relatively similar metabolic rate.
But that was estimated, right? We actually don't know what their metabolic rate is, right?
Correct, yes.
Yeah.
So that
Okay.
is part of this project, is that these are all estimates and we don't have field studies to back it up at this time.
All right, thanks.
Thank you. Any other distant sites?
Fairbanks.
Okay, we'll take questions in Fairbanks. Eric was first. Sorry.
So you just said that killer whales and salmon sharks are different sizes.
They are.
Any idea why they would both be keying on H3 ocean, like medium size Chinook?
Yeah, so we actually do have an idea about that. So until age 3, Chinook don't pass the maximum size of other salmon species, but at age 3 they start being larger than the other salmon in the ocean, so there might be some sort of herd protection before that age, and then they might start looking different, and a predator could key in on that larger prey. So that was what we came up with, because originally we were like some random age class in the middle of the ocean ages. That seems really weird.
Why wouldn't they key in on the biggest ones?
So another conclusion of this was maybe they're keying in on those first ones that they're able to see and are most abundant, and then by the time they get to those older ages, they're so not abundant, they're so few, that they really can't key in on them individually anymore, because they just don't encounter them very much. All theoretical though. Thank you.
Yeah, based on the fact that there's a lot of uncertainty about the general diet of salmon sharks, I was just curious what, for your mass balance model, you were using for the other 97% to 99% of the salmon shark diet other than sharks.
Yeah, so those diet scenarios were based on percent of total salmon that I then subdivided between salmon species, other fish, which I divided between herring, sablefish, and walleye pollock, and squid. And the other fish were divided equally between those 3 species. And squid was a mean of 5 squid species.
I was just wondering, since you have kind of shown that the age structure can change based on salmon predation, is there any indication that salmon shark abundance has increased as far as like informal or observation evidence?
So there's some informal observations that there were informal observations of increases and then some more recent report of decreases. But it's not really known. They aren't surveyed, and a lot of the surveys that catch them as bycatch catch them very, very infrequently. So, I'm not really sure. Yeah, John.
I'm just curious, like, is there anything out there on, like, for example, the migratory pathway of salmon from the Gulf out to the western coast and lower 48? Is there a correlation maybe with predation and if there's a density difference in the salmon sharks or anything like that?
That's interesting. I don't know. I think that, so salmon sharks will seasonally key into when salmon are returning to rivers. So they have been seen to be in high abundances right around where they're returning to rivers. And so that's something that has been observed. So that's happened in Prince William Sound. It's been observed off the coast of British Columbia. Has not been seen in Western Alaska. Anyone else? Thanks so much, guys, for coming. I really appreciate it.
And before everybody gets up and leaves, I'm having a celebration for Kaitlyn's accomplishments tomorrow night at my house. And if you need directions, let me know. And everybody's welcome to come over. And if not tonight, it's tomorrow night. Yeah, we'll come over tonight. In Russia.
Nice job, Keith.
Your call will be disconnected.